So, my name is Daniel Trotter, and I'm at the University of Ottawa. I'm going to tell you guys a bit about cell morphology and how it affects the response that we see to non-invasive stimulation. So, to overview a bit, non-invasive stimulation is a technique in which essentially the electric field, or sometimes the magnetic field, outside of the skull is changed with the hope of effectively depolarizing the cells in question underneath. The common techniques for this that crop up in the literature are transcranial alternating current stimulation or transcranial magnetic stimulation, alternatively called TACS and TMS, respectively. A lot of computational models that exist for this, however, use simplified neurons. The ball and stick type models that we quite often see, or leaky integrate and fire, all of the common ones. However, recently the Allen Institute has made available a selection of morphologically accurate neuron models, all of which are from the primary visual cortex. So here are 3 example morphologies for layers 5, 4, and layer 2, 3 that are pyramidal cells, and here are some additional selections that are parvalbumin cells. So again, layer 5, layer 4, layer 2, 3. So we look at them, we can kind of see that the parvalbumin cells are more bunched up and kind of bush-shaped, whereas the pyramidal cells are more elongated. So the question then arises as, what is this morphology doing? Why is it different? And how are our responses to non-invasive stimulation going to change with this morphology? Indeed, even in the same layer here, 2 example morphologies from layer 5, pyramidal cells, both pyramidal cells, but drastically different in terms of how they look and sit within space. And it's not unreasonable to assume that this might have effects. We can also then consider, beyond the actual shape of the neuron, its actual physical properties on a smaller scale. So here are 2 histograms of the mean diameter and mean length of some layer 5 cell models. So there's 5 of each. Just looking at the dendritic and axonal compartment lengths and diameters, and we can see that there are some differences. So that falls in line with existing literature of some simplified models, such as this paper by Yi et al. Out of 2017, where they actually were looking at, and they said that the Rayo base changes with the diameter and the length of the compartments within simplified models. So it stands to reason that we should expect similar changes with the morphologies that we have. How I'm going to measure it is in a quantity of mean squared error. So effectively, I'll be measuring that relative to the noise. So with the noise, so zero field applied, is this Vn term. Then for any given sweep here, just labeled V1, of an applied field at each data point, then you calculate the mean squared error, and I'll then take the mean of that as our kind of metric. So if there's more spiking, we would expect a larger mean squared error. Although alternatively, you could also get large mean squared error for hyperpolarization, and I'll get into that in a second. So it's a very overwhelming figure at first, so I'll try to break it down quickly, but essentially this top half is your pyramidal cells, of which there's 5 different morphologies. The bottom half is parvalbumin cells, also 5 different morphologies. The numbers on the bottom are the applied electric field in the simulation, which, again, computational modeling, where there is some noise. And the color schemes are then, for the purple-green one, are events in which we see depolarization, and the blue to yellow is hyperpolarization events. So the mean squared error being larger in yellow means that we're much more hyperpolarized. Larger in green means a lot more spiking. So looking at these plots, however, we can see that in this particular case, where all of these cells were artificially set to be in the kind of textbook archetypical orientation, that is, where we expect the nice kind of thin dendrite to sit along the z-axis going upwards, soma is at 0, etc. So it's your very archetypical position. But we can see that even within that position, within the morphologies, there are differences. There's a general trend of, yes, as you increase the strength of the field in the negative, you're going to get more depolarization, but how much of an effect that is quite varied. For example, there's cell 8, which these are just the numbers of the models. In the parvalbumin one, was fairly unresponsive in terms of the negative field. There's not a whole lot of difference as you change the strength of it. It would need a larger field strength to get a more sizable response. Whereas in the pyramidal cells, there's kind of this middle cluster here where we can see that they're much more responsive, and these cells are actually fairly morphologically similar to each other. The question we wanted to ask then is, what is the difference? So we take here, in this case, two orientations of the same cell. So this top row is the same orientation as on the previous slide, where it's that kind of stereotypical straight up and down orientation. This secondary row here, however, I calculate the density of the region, that is the direction in which the majority of the cell sits, is kind of an orientation, and I then reorient that part so that in this row, that densest part is sitting along the z-axis. And we can see then that putting that densest part, so changing the orientation of the cell in the field, can be optimized such that we get a stronger mean squared error, so a increase in the amount of spiking that's going on. So to summarize the kind of takeaway points, the cell response to non-invasive stimulation is sensitive to morphology, so that's an area of research that we're hoping to see a lot more work in. Ideally, we hope to be able to find a metric to group these cells together, to be able to classify them in types of cells that respond in a certain way, whether that's parvalbumin pyramidal type cells, or whether that's classifying them more on physiological properties, such as this density that we're interested in. And finally, that indeed changing the orientation of the cell in the field can be meaningfully different in terms of the response that you get. And I'll just finish with here some sources. Thanks for your attention, and I'll thank my advisor, Dr. Lefebvre, for his help on this project.